we've seen that there's a lot of plastic being discarded into the environment, and we know already a bit where it ends up and that it's dangerous for life in the ocean, but we do not really know what happens to this plastic waste. Maybe it's also coming into our bodies if the plastic breaks down into smaller particles, which are then microplastics or nanoplastics. And we want to know whether there is any health risk of these particles, and then especially to the unborn child. So I work on a digestion protocol for placental tissue. So that means that I receive actual human placentas from women who gave birth and who said that we can use their placenta for research. And then I have a protocol in the lab where I can remove all the placental tissue, and then the possible micro nanoplastics that are in there will be left over, and those we can analyze. So in our new paper we found an optimized digestion protocol for human placental tissue, and we've also found a certain recovery rate of the amount of plastic particles that we put in our sample and versus what we got out. And that's really important because we want to know if we look at any environmental particles that are already in the placental tissue, we want to know, does that link to what is actually in there? So is what we are seeing also what is actually in the tissue? I think this method can also be used for other types of tissue. I know a lot of research is right now being done on liver or brain tissue or lung tissue. I think a lot of different tissue types are being assessed for micro nanoplastics. And I think the protocol could be adapted to any other kind of tissue. And also to blood, a very similar protocol has already been used in another publication. So yeah, I think it's really widely applicable to other matrices.